All right, so a couple of years ago, I became the Vice President of ESA in charge of student affairs, and I did this not because I needed another hobby in my life, but because I got so much out of being a member of ESA when I was a student, and one of the things we really do well as a society is support our student members. So we've given out like all sorts of different travel grants and prizes and things each year, and this session is really just to recognise the amazing achievements of some of those top students. So we're going to start out today with James Fitzsimons, who's going to present the Nature Conservancy Award for Applied Conservation.
Thanks, Angela. Angela and I met for the first time in person yesterday, and I got a lovely compliment. She mistook me for the winner of this award as opposed to the presenter and funder, and I didn't have the heart to tell her that it was back in 2001 where I first presented as a student for ESA, so there you go. As Angela mentioned, I work for the Nature Conservancy. For those that don't know, the Nature Conservancy is a global conservation organisation. We work around 35 countries around the world, achieving large-scale conservation outcomes. We've been here for about 10 years now in Australia, working mainly up in the northern savannas, here in the arid lands, down in the far southwest of Western Australia, and more recently in the temperate marine ecosystems of the estuaries of southern Australia. We do a lot of work in terms of, certainly a lot of past work in helping other conservation organisations buy land, so big private land conservation purchases, increasingly helping Indigenous communities and traditional owners, creating new Indigenous protected areas on their country, including some of the largest new additions to the reserve system in Australia. We really pride ourselves, I guess, on being at the innovative end of conservation, including financing, legal mechanisms, but also for science as well. We've got around 600 scientists around the world. We're very heavily science focused, and I guess promoting science and applied conservation science is a key part of what we do here in Australia, and hence supporting this award for the last 7 years has been really important for us. So I'd really like to thank the ESA. It's been a great partnership between TNC and ESA over the last 7 years, and particularly thanks to ESA staff and the judges for this award. It's been another strong year. We had 15 applications this year for the Applied Conservation Award, and it was such a strong field that we gave a couple of special commendations to Lauren Young and to Michelle Freeman. But I'm really pleased to announce that the winner for this year's award, 2014, is Lachlan Feder of the University of Wollongong. He was selected to have submitted the best research application for his PhD research, the conservation of soft sediment fishes, the vast unknown. Now, Lachlan's research focuses on the fish that live in the soft sediments, predominantly sand, which covers the seabed for most of the Australian waters. He'll be investigating the effectiveness of conserving this group of species in marine protected areas, which is an incredibly topical issue at the moment. Surprisingly, there's been very little research, both here in Australia or internationally, on this subject. And Lachlan's going to be using tag tracking technology in Jervis Bay Marine Park, as well as baited remote underwater video stations in both no-take sanctuary zones of the marine park, but also in recreational fishing zones and also outside of the marine park in commercially fished areas as well. And what this technology will do is actually help him to determine if common soft sediment fishes like blue-spotted flathead and eastern school whiting inhabit specific areas and whether they return to the same areas after migration. So without further ado, I'd like to welcome Lachlan to the stage to receive the 2014 TNC Applied Conservation Award. Lachlan will be presenting the findings, I guess, of his research this time next year at the Adelaide ESA conference. And so I'm really pleased to welcome up to the stage Catherine Berry now. Catherine's from James Cook University and the Australian Institute of Marine Sciences. Catherine won last year's award for her research on another really topical issue. I'll tell about it in a minute. It's a really topical conservation issue and one that's in the press constantly at the moment. And she's going to present some of her findings to date. The title of her presentation today is on coal spill scenario, acute and chronic effects of coal dust on a reef building coral, seagrass and fish species. Catherine.
Thanks very much. So Australia is currently the largest exporter of coal in the world, and Queensland actually holds the 4th largest global reserves of coal. Approximately 97% of the coal that's mined in Queensland is exported through the Great Barrier Reef via the ports that are shown in the figure on the right. In 2012, 156 million tons of coal was exported through the Great Barrier Reef, and this number is forecast to increase 2- to 3-fold over the next few decades, reaching approximately 300 to 450 million tons by 2025. And as you can see in this figure, there are some graphs with every port showing the current tonnage of coal that's exported on the left-hand side and the bright blue bars. Well, the dark bars represent the proposed tonnage with the forecast exports. So in order to facilitate increased exports of coal through the Great Barrier Reef, ultimately there needs to be an increase in coal vessels. So this graph here was created by Grabumpa, and it shows coal vessel projections in the Great Barrier Reef over the next few decades. Now, it must be noted that these values are considered as the upper limit, but the forecast shows significant increase in coal vessel shipment through reef waters. Now, this is particularly concerning because the Great Barrier Reef consists of over 3,000 individual reefs, in addition to the islands. It's a very complex system to navigate through. So with this significant increase in shipping vessels, there's the likelihood for increased ship grounding, such as the Sheneng 1 coal carrier vessel, which grounded in 2010 off Douglas Shoal in the southern Great Barrier Reef. It was transporting 68,000 tons of coal at the time of its grounding. Luckily, in this scenario, no coal was released. However, this picture here shows the MV Smart, which recently grounded off South Africa just last October while it was carrying 110,000 tons of coal. During this grounding and salvage operation, 60,000 tons of coal was released into the marine environment, and you can see this in the black plume that's emanating from the vessel and also in the left-hand corner of the picture. Oops. Sorry. So just down here in this area. So unfortunately, there are currently large knowledge gaps pertaining to the risks associated with coal dust in the tropical marine environment. Therefore, we're currently unable to competently assess the risks of coal dust to tropical marine organisms. So this brings about the question, what are the acute and chronic effects of coal dust on tropical marine biota? So what we do know at the moment is that there are 4 main features of coal dust that may result in adverse biological effects to marine organisms. The first is the physical presence of large amounts of coal dust in the water column, which may result in smothering and abrasion of sessile benthic organisms, as well as clogging of feeding and respiratory organs. The second is the attenuation of light in the water column by the particles, and this will ultimately have adverse biological effects on photosynthetic organisms. The third and the fourth features are the release of inorganic and organic constituents of the coal itself, such as trace metals and hydrocarbons, such as polycyclic aromatic hydrocarbons, both of which are potentially toxic to marine organisms. So the risk of any contaminant is a factor of exposure, so the concentration and the duration times the impact to relevant species. So in order to investigate the potential risks of coal dust on tropical marine organisms, I recently conducted an experiment at the National Sea Simulator at the Australian Institute of Marine Science. So I investigated the effects of coal dust on 3 organisms, a reef-building coral, Acropora tenuis, a reef damselfish, Acanthochromys polyacanthus, and a seagrass species that's commonly found in port areas along the coast of Queensland, Hologili uninervis. And I exposed these organisms to 5 different concentrations of coal dust in triplicate tanks. The concentrations were 0, 25, 60, 80, and 175 milligrams per liter. And I exposed these organisms for a duration of 28 days, but I had sampling points at 4 days, 14 days, as well as the 28 days. Now, this was a flow-through system, so there was constantly fresh seawater being added into the tanks and wastewater being flowing out. So in order to compensate for this loss, I had 6 header tanks on a mezzanine above the experimental tanks. They were 200 liters, and they contained highly concentrated stock coal solutions, which were continuously kept in suspension. They were set to a timer, so 10 times an hour, a pre-calculated volume of the calculated volume of the coal solution was pulsed down into the experimental tanks, and this allowed me to maintain the desired concentrations in the experiment. So during the experiment, I collected data for various biomarkers such as growth, photosynthetic efficiency, mortality, and respiration. And I also collected water quality parameters such as light attenuation, total suspended coal, formerly known as total suspended solids, pH, temperature, sedimentation, dissolved O2, turbidity, and trace metals. Now, due to the time limitations of this presentation, I won't be able to go over all of these results with you today. However, I have chosen one biomarker from each organism. And I'd just like to point out the fish in this image. As you can see, they are tagged with a fluorescent dye. So there were 10 fish in each tank, and they were individually tagged with a distinctive color of fluorescent elastomer, and this allowed me to measure individual growth in each fish over time, which increased the power of our data. So just to start with the coral, some of the coral results. Here, this graph shows you coral mortality. So on the x-axis, we have the 5 different concentrations at each respective time point, and on the y-axis, partial mortality in percentage. So this was the percentage of each nubbin that was either completely healthy, exhibiting partial or full mortality. So the light gray bars are depicting healthy nubbins, while the light blue bars show partial mortality, and full mortality is represented by the dark blue bars. So as you can see here, partial mortality kicks in at 25 milligrams per liter or greater coal dust at 14 days. And the median lethal dose, which is the dose required to kill 50% of a test population, was 25 milligrams per liter at 28 days. And the pictures on the right just give you an example of what we were observing in the nubbins over time. So some of the nubbins were exhibiting slight bleaching of the tissues, while others gradually were exhibiting tissue sloughing, complete tissue sloughing, and full smothering of the coral nubbins. I used leaf extension in millimeters per day as a proxy for growth in the seagrasses. On the x-axis, we have the exposure days, and on the y-axis, the growth per day. So as you can see, at about 4 days, nothing much had happened. However, the seagrasses exposed to the 3 highest concentrations of coal dust did exhibit growth inhibition that was significantly different than the relative to the controls at both 14 and 28 days. And the pictures on the right just show you an example of the seagrasses in the control treatments compared with the seagrasses in the coal exposures. So that seagrass on the right side was exposed to 175 milligrams per liter of coal. And as you can see, the coal is quite sticky. It stuck right to the epiphytes and as well to the seagrass leaves. This graph here represents juvenile reef fish growth rate. So once again, on the x-axis, we have the growth, the exposure times in days, and the proportional growth on the y-axis. So that was the percent increase in size from the start of the experiment. And as you can see, which is consistent with the other results, at 4 days, we're not observing many differences between the treatments. However, at both 14 and 28 days, there was significant growth inhibition in all fish exposed to coal relative to the controls. And as you can see here, the fish that were exposed to any coal dust concentration only had about a 10% increase in size from the start of the experiment in comparison to the controls, which increased in size by about 20%. So the controls grew double the amount of the fish exposed to the coal dust. And the picture here just shows you the differences in physical appearance between the juvenile fish in the control treatments as well as those in the coal treatments. So just to summarize these preliminary conclusions, in general, the organism showed little response to acute coal exposure at 4 days. However, in concentrations 25 milligrams per liter or greater, there was partial mortality observed in corals within 14 days of coal exposure, and significant growth inhibition in fish within 14 days. In higher concentrations, 60 milligrams per liter or greater, there was significant growth inhibition in the seagrass within 14 days, and 100% mortality in the corals within 28 days. So what we're really hoping is that this data will provide industry and regulators with scientifically rigorous information to improve environmental impact assessments, risk modeling, and the management of coal shipment through the Great Barrier Reef. And this image here is just taken from the RG Tana coal loading facility in Gladstone. Just to give you an idea of the magnitude of coal that's currently waiting to be shipped through the GBR, and this will just keep getting larger over time. So I'd just like to acknowledge the funding bodies for this research, the Ecological Society of Australia and the Nature Conservancy. Thanks very much. I'd also like to thank the Australian Institute of Marine Science, AIMSA JCU, and the Great Barrier Reef Marine Park Authority. I'd also like to give a special thanks to my volunteers. Without their help, this experiment would not have been possible, as well as the support of the CSIM staff and the staff at Marfew for helping with the fish rearing. Thanks very much.
Thanks very much, Catherine. We've got time for 2 quick questions from the floor, if anyone wants to fire away. Yep, over here. Sure.
What were the concentrations like in your experiment compared to what you might find in nature?
So the question for those down the back, what were the concentrations like in the experiment compared to what you'd find actually in nature?
Thanks. That's a really good question. I'm asked it all the time. So there's unfortunately no data at the moment of concentrations of coal dust in spill scenarios, and not a lot of environmental monitoring was conducted during the MV Smart grounding, unfortunately. Next time I plan to fly to a grounding site. However, compared to what other experiments have looked at, there hasn't been a lot of research done in tropical areas. However, research has been done in temperate areas on the effects of coal, and other people have looked at concentrations ranging from about 3,000 milligrams per liter to 10,000 milligrams per liter. So the concentrations I looked at are actually much lower than that, and they still showed these severe effects.
Any others? So the question was prospect for recovery.
Well, I guess it depends on the organism. With the corals that exhibited full mortality or tissue sloughing, the chances of recovery are quite low for those impacted corals. With regards to the fish, although we did see inhibition in growth, there were very few mortalities throughout the duration of the experiment, so I would have more hope for them in the future. Unfortunately, due to time constraints, as the CSIM is a very popular facility, we didn't have the chance to do a recovery period. I will be doing some more experiments next year, looking at more of a pulsing acute or a chronic response with lower doses, so we'll have a better idea of how certain organisms, specifically corals, will be responding to smaller amounts of coal and how that affects their energetics over time. So maybe next year I can give you some more information on that.
Thanks very much, Catherine. That's it for the T&C Award for this year.
Thank you.
So our next award for the morning is the Jill Landsberg Trust Fund Scholarship. And if I could have Daniel Bateman up here to receive his award. Sorry. Congratulations. And We have also last year's winner of the Jill Landsberg Trust Fund Scholarship here, Jorge Ramos, and Jorge's title is Population Genetic Signatures of a Recent Climate-Driven Marine Range Extension. Thank you.
That's for what? This one? And other one. Hi, morning. So many marine species are changing their patterns of geographic distribution, abundance, and phenology in response to oceanic warming. This is because they are tracking their preferred thermal environments. However, the magnitude in these responses is highly variable, probably because there are many variables involved in these processes. Recent studies have shown that between 25 and 80% of these changes in distribution are consistent with the direction of climate change, including oceanic warming. So these changes in distribution result in changes in the structure and function of the ecosystems that have important consequences for social and economic activities. So it's very important that we know what species are likely to cause to undertake these changes in distribution caused by oceanic warming, so we can better anticipate to these potential impacts. So in this study, we're studying the range extension of the common Sydney octopus, Octopus tetricus. This octopus has a planktonic larval phase that lasts about 60 days, and it can be transported up by oceanic currents. The historical distribution of the octopus is from southern Queensland to southern New South Wales, but in the last decade, we have found it in Victorian and Tasmanian waters. This range extension has been associated to the strengthening of the East Australian current that has actually extended approximately 350 kilometers further south, and apparently has brought more than 70 species into Tasmanian waters. So yeah, my thesis, this is for my PhD project, and my thesis is the examination of the life history characteristics and the population dynamics of these species and how they influence this range extension process. But today, I'm just gonna talk about the genetics component of my project. So we aim to answer the following questions. What is the population genetic structure of Octopus tetricus along the entire distribution? What is the gene flow between historical and extension areas? Where are the source populations located that contribute to the range extension areas? What is the genetic diversity of the range extension versus other population components? Are there any bottleneck, founder effects, or demographic effects that might affect the population? And finally, how these genetic signatures influence on the persistence of these species in new areas. So we collected samples in 3 sites along the historical distribution. These are Namboka Heads, Swansea, and Merimbula, and in 2 sites at the range extension. These are Malakuta and northeastern Tasmania. And we found 2 morphotypes. Morphotype 1, we found it along the entire distribution. This has thin, long arms and a mantle. The mantle is like the head. So yeah, that's the mantle. So yeah, sorry. So yeah, the mantle is small in proportion to the rest of the body. And the morphotype 2, we found that only in Tasmanian waters, and this has thick, short arms and a mantle that is big in proportion to the rest of the body. So there seems to be some phenotypic plasticity of these species at the range extension. So we collected a piece of tissue, then we extracted the DNA, and we amplified 7 microsatellite loci with fluorescent tags. The PCR products were separated by capillary separation, and we genotyped these products. We examined the population genetic structure using 2 approaches, and we estimated genetic diversity by measuring the number of alleles, number of private alleles, allelic richness, observed and expected heterozygosity. We also estimated genetic connectivity using the FST values, and dispersion and migration rates, and self-recruitment. We test for any evidence of a bottleneck effect, and we also estimated effective population size. All of these parameters were estimated for the historical distribution and for the range extension, and for each site. So important to mention that all of these microsatellites were in Hardy-Weyber equilibrium, so good. Population genetic structure analysis, we identify 5 clusters. So you can see the clusters in this figure, And you can tell that, well, the colors of each cluster correspond to the colors in the pie charts, and these pie charts indicate the percentage of individuals from each side that corresponded to that cluster. So we had these 4 clusters that were very close, and they were common all along the entire distribution, but there was also a distinct cluster, the TA2, I call it this way. So yeah, the distinct cluster TA2 that was mostly comprised from Tasmania, from individuals from Tasmanian waters. So yeah, we have one group along the entire distribution and one group, one distinct group in Tasmanian waters. FST values were low. This is an indication of high gene flow between sites. Still, there was some level of genetic differentiation between the distinct Tasmanian cluster from the rest of the population, and there was some level of differentiation between the rest of the individuals from the ranch extension from the historical distribution. Dispersion and migration rates were asymmetric. This is that most of the migration or dispersion occurred from along the historical distribution towards the ranch extension. But when we compare among sites, there was migration from all sites to all sites. So in this table, maybe just have a look at Malakuta. That's the site that had the greatest migration rates towards the historical distribution and towards the ranch extension. And also there was high self-recruitment mostly for the sites at the ranch extension, but in particular for the distinct Tasmanian cluster TA2. So we have that the ranch extension area received immigrants from along the historical distribution area, but Malakuta was the main source of migrants, and then there was moderate to high inflow between sites. Genetic diversity did not decrease at the ranch extension. This was unexpected because usually species that undergo changes in distribution, they experience or suffer a series of bottleneck effects that decrease the genetic diversity, but this was not the case. So genetic diversity was sustained at the ranch extension area. And this, according to the literature, this is something like a rapid demographic expansion, which makes sense. There was no evidence of a bottleneck effect. In this table, I'm showing you the p-values for each of the tests that I did for each site and area. So there was no evidence of bottleneck effect. And so all seems to be positive for the ranch extension of this species in the new areas of its distribution. But when we estimated the effective population size, we found that the effective population size at the ranch extension was very little compared to the historical distribution. According to the literature, an effective population size of 100 individuals can avoid inbreeding depression. This is the case of the ranch extension. And an effective population size of 1,000 individuals can retain evolutionary potential. So the way I see it here, well, yeah, the effective population size is very little at the ranch extension area. And the way this component of the population might sustain the genetic diversity might be through the gene flow, high gene flow from along the historical distribution. So this high gene flow helps sustaining the genetic diversity and the adaptive capacity at the ranch extension area. So in summary, we have that there might be two subpopulations, one along the entire distribution, one in Tasmanian waters, that there is moderate to high gene flow between sites, and this gene flow comes from all along the entire distribution. So think of the gradient of environments and habitats from Queensland to Tasmania, and individuals living in those different environmental conditions. So those individuals in different environmental conditions might have different gene pools, and when they mix at the ranch extension area, then they sustain the genetic diversity in these new areas, and they might increase the adaptive capacity. So genetic diversity did not decrease at extension areas, and there was no evidence of a bottleneck effect, and effective population size decreased, or at least was little at extension areas. So how these genetic signatures influence the persistence in new areas? Okay, we have two scenarios here. A positive one above the gray line and a negative one below the gray line. And think of these triangles as the magnitude of each of these factors. So the white side of the triangle is great magnitude, and the narrow part of the triangle is little magnitude. So these are the factors that we examine in this study. So we have, when populations that have high gene flow, high diversity of source of migrants, high genetic diversity and high phenotypic plasticity, and that do not suffer the effects or a great magnitude of the effects of bottlenecks, yeah, you know what I mean, or demographic effects, then these populations might have better chances to prevail in new areas. So what happened with Octopus tetricus, it went did really well for most of these factors, but given the effective population size, we are not sure if there is a demographic effect that negatively affects the population at the range extension, then the octopus might not succeed in the new areas, but if there is no negative demographic effect, then the octopus might have really good chances to prevail in the new areas. So there are many other factors that influence the range extension of marine species, and it would be great if we could have like the list of all these traits and how they might influence this range extension, and then this way we could better anticipate these potential impacts. So I'll just leave it there. And finally, I'd like to thank the institutions and people involved in the collection of samples, ESA for the invitation, and well, the 2013 Jill Landsberg Award, because without this support, I wouldn't have been able to conduct this study. And that's it. Thanks.
Thank you. We have time for some questions for Jorge.
How strong is the evidence that the octopus wasn't in Tasmania historically?
Can you repeat the question?
Yeah, so how strong was the evidence that the octopus was not in Tasmania before? Yeah, so there are census. For example, you have the Reef Life survey that they have census all around Australia and around the world. There are many other works that have described the distribution of this octopus, and they didn't identify this octopus at least in Victoria and Tasmanian waters after 2000. So there's, also through the fisheries, there is a fishery of octopus in Tasmania, and they started to catch this octopus after 2006, approximately. So yeah, it seems to be like it's happening, yeah. Yeah, so if there is any, sorry, what's that? Yeah. Yeah. So yeah, that's interesting. So yeah, gene flow was high between sites, but was very limited for the distinct Tasmanian cluster as well. And there was high self-recruitment. So what I'm thinking here is that there is high gene flow to the rest of the individuals in Tasmania that were not part of that distinct Tasmanian cluster. And the distinct Tasmanian cluster is becoming, what I'm thinking here is that it's becoming a separate subpopulation that is self-sustained. And yeah, so it's becoming something apart. And yeah, the phenotypic plasticity, well, that's something that it was a surprise. We didn't have any record of this, and we identified the species. So yeah, so I need to start to do more analysis, like morphometrics and stuff, to make sure that there is a real difference. So yeah, but I haven't done, so I will soon. Yeah.
Okay, we'll go one more, I think.
Do you know what the larval life of the species is, and how far a larva is likely to be able to travel in their currents?
Yeah. So how large is the larval stage and how far it can travel? Yeah, so there is no real estimation for this species. The estimation that I said, 60 days, it's based on a very closely related species. How far can they travel? Well, it depends on the dynamics of the East Australian current, but not also on the current, also on the coastline, eddies, fronts. There are many variables affecting the dispersion of these species. So the dispersion might be very patchy, actually. And So, for example, I know this is basically terrestrial conference, and I know you use the landscape genetics, and so now they are proposing in marine life like to use like seascape genetics, including all the life history characteristics, oceanographic characteristics, and other ecological variables. So, yeah, it would be hard to tell. Yeah, I mean, I made an estimation, and according to my numbers, it could move from Queensland to Tasmania, but it's just very theoretical. So, yeah, I don't know. Yeah.
All right. Thank you very much.
Thank you.
So I should have said more about the Jill Landsberg Trust Fund Scholarship, actually. It is, of course, in honour of Jill Landsberg, and it is a fund that people can donate money to. So if you don't want to give the money to your own students, no. Seriously, it is really important for our students to be able to get these grants. It gives them funding for their research, but more importantly, it gives them an amazing high-profile prize on their CV that's going to help them get ahead and get jobs and things, which we all know is something tricky in the current climate. Okay, the next award, can you find my slide with Wily? Is the Wily Fundamental Ecology Award. And this is something, you will have noticed that we have a lot of Wily awards. So Wily publish Austral Ecology, and they are really generous in terms of providing all sorts of support to the society, particularly in student awards. So this is one of those. And this year we have a presentation from Peter Lowe from Sydney University. And I don't have your title, so you're going to have to say it yourself, Peter. Sorry. Welcome.
Thanks. So my talk's on the effects of predation risk on insect herbivore foraging. The process of acquiring food is necessary for survival, but often places an animal at increased risk of predation. Animals can potentially decrease their predation risk through various anti-predator responses, though these often come at the cost of decreased food acquisition. This conflict between foraging and avoiding predation, which represents one of the most fundamental trade-offs in ecology, could be optimised if animals are capable of assessing short-term changes in predation risk and matching their investment in responses to the current level of risk. So perceiving changes in predation risk requires the use of cues. These can be direct, where they're produced by or related to the presence of the actual predator, or can be indirect, where they're correlated with the probability of encounter. And they can be in the form of chemical, physical, visual, or auditory stimuli. So the information gained from these cues can be processed into an assessment of the level of risk to produce a response from the animal that can be behavioural, physiological, morphological, or life historical. So there's a lot of evidence to show that animals are capable of assessing changes in predation risk in this way. And while a lot of the research has focused on vertebrates, there's also growing evidence for invertebrates. For example, we recently showed that caterpillars having experienced a failed predator attack, a direct physical cue of predation risk, subsequently alter aspects of their foraging behaviour to minimise future risk. This study and many others, however, have typically focused on one predation risk cue at a time and on response types taken separately. In particular, there's been a focus on behavioural responses, while we know relatively little about physiological responses, especially when it comes to invertebrates, or about how different response types are integrated. So my aim, therefore, was to test the combined and individual effects of a direct physical and indirect chemical cue of predation risk on the behaviour and physiology of an insect herbivore. As a model species, I used caterpillars of the black slug cup moth, Dorotifera castor, pictured on the left, and this is an endemic generalist herbivore that feeds on trees in the family Myrtaceae. So I collected caterpillars from Sydney Harbour National Park and used them in experiments when they were in their 3rd to 6th instar. The predation risk cues that I tested included a direct physical cue in the form of a simulated predator attack and an indirect chemical cue in the form of a mortally wounded conspecific. So I simulated predator attack by gently pinching a caterpillar's body 4 times using soft forceps, while conspecifics were wounded by puncturing their body with sharp forceps. Forceps are commonly used to simulate the peck of an insectivorous bird or the mandible bite of an arthropod. So I tested the responses of caterpillars to these cues in 2 full factorial experiments. The first looked at effects on behaviour and food intake, while the second looked at effects on metabolism. So to investigate the effects of these predation risk cues on behaviour and food intake, I conducted 3-hour feeding trials. This involved placing caterpillars individually onto Angophora floribunda leaves held in vials of water, and the caterpillars were allowed to settle for 5 minutes before the treatments were applied. So a caterpillar was either pinched 4 times or left undisturbed and was exposed to one of the 3 chemical cue treatments. So either a wounded conspecific, a non-wounded conspecific, or a no conspecific control. I measured any immediate behavioural responses of caterpillars to attack for those exposed to the physical risk stimulus, and then at the end of the trial I calculated leaf area consumed and the amount of food assimilated. So the feeding trials were conducted in a constant temperature room set at 23 degrees and blocked by chemical cue, and I had 40 caterpillars per physical chemical cue treatment combination. So the caterpillars showed a range of immediate behavioural responses to attack. The most common was to avert their stinging hairs. So this picture just shows a couple of caterpillars, and you can see in the individual on the front left that the 4 rosettes of stinging spines are retracted while they're exposed in the individual next to it. Other common responses to attack included to stop feeding and to walk away, and caterpillars often showed multiple responses to attack. Only a few individuals from the no conspecific control treatment did nothing in response to attack. Overall, however, we found no difference in the suite of immediate behavioural responses to attack between the chemical cue treatments. What I did find, however, was that in response to the physical cue of predation risk, caterpillars significantly decreased food intake and assimilation. So decreasing feeding is a common response to increases in predation risk and is expected to decrease future risk of predation, for instance, by decreasing the availability of visual cues like feeding damage and movement associated with feeding, and chemical cues such as plant volatiles, which natural enemies of insect herbivores often exploit to locate their prey. So for the second experiment, which aimed to look at the effects of these predation risk cues on metabolism, I exposed caterpillars to the same set of physical and chemical cues and measured rates of oxygen consumption with a 5-box-3 oxygen system. So this method represents one of the latest advancements in respirometry for small animals, and it uses a fibre optic cable seen in the picture to read the reflectance of a fluorescently coated oxygen sensor spot. So I placed caterpillars individually into small vials that also contained the oxygen sensor spot and a square of paper toweling with a drop of water for humidity and the chemical cue. So for caterpillars exposed to the wounded conspecific, the paper toweling had been rubbed against an injured caterpillar. For caterpillars exposed to a non-wounded conspecific, the paper had been in contact with a non-wounded caterpillar, while for the no conspecific control treatment, a clean piece of paper toweling was used. And I applied the physical cue by pinching half of the caterpillars 4 times before the vial was sealed. So I measured levels of oxygen in the vial immediately after sealing the vial at 15 minutes, 30 minutes, every half an hour for 3 hours, and again 20 hours later. And from this, I measured mass-specific rate of oxygen consumption for each time interval. This picture just shows the whole setup. So in the front left corner, you can see the caterpillars in their vials. Then there's the 5-box-3 system with the fibre optic cable and the computer program recording the readings. Again, the experiment was conducted in a constant temperature room blocked by chemical cue, and I had 20 caterpillars per physical chemical cue treatment combination. I calibrated the equipment daily with nitrogen and air blanks. So I found significant effects of both the physical and chemical cue of predation risk on metabolism, though these effects were independent and occurred on very different timescales. So in response to the chemical cue of predation risk, caterpillars showed an acute response, where within the first 15 minutes, individuals exposed to a wounded conspecific had significantly elevated rates of metabolism compared to caterpillars exposed to non-wounded conspecifics. Such increases in metabolism represent a common response to predation risk seen across taxa that typically occurs as part of an induced physiological stress response. It was interesting that the metabolic rates of caterpillars from the no conspecific control treatment were intermediate. A possible explanation for this could be that being alone was perceived as partial vulnerability. So in contrast to the short-term acute response of caterpillars to the chemical cue, the physiological response to the physical cue of predation risk occurred only over the longer term. So only by 20 hours did caterpillars that had experienced a simulated attack show significantly elevated rates of metabolism compared to undisturbed caterpillars, suggesting a chronic accumulation over time. So some conclusions. I found that the effects of these chemical and physical cues of predation risk were independent, since caterpillars responded differently to the cues and the responses occurred on different timescales. So in response to the physical cue, caterpillars reduced feeding and assimilation and showed an increase in metabolism over the long term, while in response to the chemical cue, caterpillars showed a very short-term increase in metabolism. While the behavioural response to decreased feeding following attack likely decreases future risk of predation, the physiological response that we observe to both the physical and chemical cue likely increase the costs of this behavioural response, for instance by increasing basal energy demands and the energetic costs of anti-predator activities. So my results provide further evidence that caterpillars can show sophisticated responses to predation risk and also highlight the importance of an integrative approach to achieving a better understanding of the potential benefits and costs of anti-predator responses. Thanks.
Excellent. Do we have questions for Peter?
Yeah, I couldn't control for the disturbance of placing them on the plant, but I used a soft paintbrush to minimise any disturbance, and I allowed them time to settle. And I guess it's a gradient of disturbance, so the ones which were pinched would have received like that extra, very obvious disturbance.
Any other questions? Can you repeat the question once? Don?
What do you think of the consequences of these changes in metabolism for their, when they turn into butterflies and reproduce?
So the question was, what could be the consequences of these changes in metabolism? It's a good question, and I'd like to do further research over a longer term to see what the consequences could be, because these were obviously very short-term responses. But I think in the wild, the predation risk is pretty high, so it would be a chronic sort of impact on them. So I think it could have a pretty dramatic effect.
One more.
Yeah, you showed a picture of a cuckoo there which eats those kinds of caterpillars. But I was just wondering, is very much known about how those little rosettes of hairs would actually deter a bird or a bug that was about to eat a caterpillar?
No, not a lot is known. I guess it's probably not that effective against some insects, because I've seen them being eaten by bugs and they just attack them from behind where the hairs aren't there. But they, I guess, should have some benefit, otherwise they wouldn't be there, I suppose. Yeah.
All right, so thank you, Peter. So next up, we have a brand new award called the Next Generation Ecologist Award, and that's going to be presented and introduced by your president, Nigel Andrew.
Thanks, Ange. So as you've heard this morning, we've had some great PhD award winners and doing some great work. And in fact, most of the talks you hear this week by PhD students are really fantastic. And I think ESA does a great service in mentoring and supporting our student members. And I also think as president, we need to also do more to support our early career researchers. One of the key aims of the Ecological Society of Australia is to mentor and encourage early career researchers and to develop strategies to engage and improve the support for these key researchers, both in Australia and also the work that they do worldwide. So today, as President, I'm really excited to announce a new award to acknowledge the work of early career researchers, and we've called it the Next Generation Ecologist Award. And this award is to encourage the best and the brightest of our early career researchers to present a plenary session, and the Society will also be paying for their accommodation, their flights, and the cost of registration to come and present at each of the annual conferences. So the awardee every year will actually be giving their vision of ecology and where the researcher thinks ecology is heading or should be heading into the future in their particular area of ecological research. So to promote this award and to give it and to actually show you some of the great work that early career researchers are doing, I've invited Assistant Professor Rachel Standish from the University of Western Australia to formally promote this new award. Rachel has a broad interest in ecology and its application to the management and restoration of native ecosystems. Her research is grounded in theory, but is driven by an interest in developing practical outcomes for restoration in a rapidly changing world. I think Rachel does some great work, and I think she's a great way to introduce this award. So, and also Rachel will be presenting her vision of ecology to you all today. So I'd like to welcome Rachel up to the stage to actually promote the award. Thanks, Rachel.
Right, thanks Nigel. So as Nigel said, he's asked me to talk about my vision for the future in restoration ecology, the field I've been working in the last 10 years or so. But before I do that, I'm going to look at some of the recent advances in my field. It was Anthony Bradshaw, one of the founding fathers of restoration ecology, that said restoration should be an acid test of our understanding of ecology. Today I'm going to look at the flip side of that question, which isn't delved into quite so often, and that's what does the practice of restoration afford us in learning about ecological theory. So when we're out there fencing our plots from herbivores, as my student Christine Allen has done here as part of a rare species translocation, when we seed, when we plant, what do we learn about the ecology of our systems? I'm going to offer you my uniquely southwestern Australian perspective. It's where I live and work, and it's these ecosystems which I understand more than others in the world. However, I also think they're more broadly, they offer us the chance to test some theory, and that's because the special traits about the flora and the landscape which make them good test beds to challenge theories, particularly those developed in the Northern Hemisphere. Steve Hopper summarised some of the special traits about the landscape in southwestern Australia as part of his OCBIL theory. So OCBIL stands for Old Climatically Buffered Infertile Landscapes. And Steve came up with a few ideas about the ecology of these landscapes. He didn't get it all right when he talked about restoration, and Richard and I were quick to point this out. However, he basically said it was going to be easy, and we don't agree with that. There were some ideas that resonated with us, and they were firstly that the flora have very limited dispersal, and this is thought to have evolved as a response for local persistence in the ancient landscape. The landscape is very ancient. Some of the rocks in this middle photo could be up to 3,000 million years old. So the soils are very ancient, and the native plants have evolved these specialised ways of living in these soils, such as the cluster roots shown on this Hakea species in the bottom there. So overall, lack of dispersal among the flora and very sensitive to any changes in soil with restoration. What Steve neglected to mention was the social context of restoration, and that's important because it determines how much we learn about the ecology. So in Western Australia, we have three primary land uses: mining, urbanisation and agriculture, and they have these different imprints on the landscape. So you end up with landscapes that look a bit like this, where the pink is the native remnant vegetation, and the yellow is the affected, the landscape affected by the land use. And also the social context sets up the goals and the money available for restoration, which in turn affects how much we know about the ecology. So essentially we know quite a lot about Jarrah forest being affected by mining, and considerably less about the systems impacted by urbanisation and agriculture. I'll walk you through my research that's occurred in 4 different study sites. The first is in the central wheat belts, Calabarin, that heavily cleared landscape down the middle there. The second in the Jarrah forest, just south of Perth. And the third and fourth, a couple of oldfields restoration sites further south. And these study sites in a bit more detail. So Calabarin was essentially a study that Richard Hobbs, Vicky Kramer and I did to understand the factors that might affect why oldfields or abandoned farmlands around the Calabarin area weren't recovering back to the native historic woodlands up to 30 or 40 years after the farmer walked off the land. The second in the Jarrah forest, looking at how Jarrah forest communities reassemble after bauxite mining disturbance has ceased. The third is Ridgefield. It's a farm. Ridgefield is a farm owned by the University of Western Australia, where we've set up a large-scale field experiment. And Peenyup, which is part of the Gondwana Link initiative down south. It's a carbon-funded biodiverse carbon project, which we've been following since it was seeded in 2008. So the first theory that I think has the potential to be enhanced by restoration practice is around community assembly, and I'll walk you through a few examples of those from the Jarrah forest and also from those other systems I just introduced. The second, I think, is ecological thresholds, particularly in the ecosystems that we've trashed in the wheat belt of Western Australia. I'll talk a little bit about this. If you want to hear some more, Richard's going to talk about ecological thresholds some more on Thursday. I also think restoration settings offer really good potential for us to explore the links between biodiversity and ecosystem function, and I'll talk about that a little bit. And finally, I look to the future at what our next generation of ecologists might start to explore. So firstly, community assembly, and the filter model has been really popular as a ways of restoration practitioners to conceptualise what they might need to do to intervene to get their desired end goal. This particular model was developed by Jennifer Funk and colleagues based on an earlier one developed by Paul Keddy. Very popular. It's one of Jennifer's most highly cited papers. It basically allows us to see if we increase or decrease the mesh size of those different dispersal abiotic and biotic filters, or change the species that are introduced to a site, how that might determine the final outcome. We've certainly got evidence from our research that each of these filters can be important. So, for example, on oldfields, Vicky Kramer and I put out seed traps at differing distances from the seed source to look at dispersal limitation. Richard Harris and I followed the fate of ant-dispersed species, and in both cases that seed dispersal was limited to distance from the seed source. We also have evidence from the Jarrah forest that the abiotic filters are very important. So, as you might know, soils in Western Australia are naturally phosphorus limited, but there's acknowledgement that we have to add a little bit of phosphorus to kickstart Jarrah forest restoration. But what this work shows, and there's increasing phosphorus levels in the fertiliser across the x-axis there, is that if you add too much, you just get a higher contribution of those weedy and ephemeral species. And in fact, this compositional similarity of those plots with increasing amounts of phosphorus as you move, similarity to the benchmark, which is the unmined, unfertilised Jarrah forest, it decreases with increasing phosphorus. We also have evidence from the Peenyup site that abiotic filters are important. So this is data that my student and Richard's student, Lauren Hellett, collected. She followed or tracked the number of seedlings to emerge from areas that were seeded by Greening Australia and found this distinct relationship with soil type. So emergence was higher in the sandy soils. Lauren then came back after the summer drought to look at what happened to these seedlings. Of course, the summer drought is a very strong filter to seedling establishment in this Mediterranean climate area, and found that seedling establishment was much higher in the clay soils. And so overall, that last figure C there shows that those two relationships essentially cancel each other out. So what Lauren's work really highlighted was the point that filters are dynamic through time, and unless we track those changes in time, we're going to miss them. So that was her contribution to theory. We've also looked at biotic filters at Peenyup, and this was largely enabled because the seeds were seeded into rows, which enabled us to collect spatially explicit data and track the fate of individual seedlings. At 5 years, the restoration looked a bit like this, and we were able to lay the tapes out in rows each year and track the survival of those different species. We hypothesised that facilitation might be important. Facilitation is very important in other Mediterranean climate ecosystems, but so far the evidence for them being important, or facilitation being important in southwest Western Australia is fairly limited, at least in part because people haven't looked. So we hypothesised that the yate planted as the carbon species might provide shade to the seeds coming up from the seed bank. Also that the prickly species might provide protection from herbivores, and finally that the legumes inputting the nitrogen might be important for facilitation. But essentially we found no evidence for any of those mechanisms, and this is work that George Perry and Nancy Shackelford have helped me put together. So what George did with this K function on the y-axis there was look for evidence of clustering at different distances between nearest neighbours. And George tells me, he understands this more than I do, that the red line should be above, out of that cone if we were to look for evidence of facilitation, and it's not. Okay, so skipping to the next ecological sort of theory that I think has been informed by restoration practice, and that's our understanding of thresholds. And this is predicated on the work of Buzz Holling, who first introduced the concept of resilience to the ecological literature in 1973. His basic idea was that if we disturb systems and they recover back to their pre-disturbance state, then that ecosystem is resilient. And if they don't recover and collapse into a different state, then they're not resilient, and there's a threshold that separates those two states. Ecologists are fond of summarising these thresholds as in these sort of state and transition models, and this is one that a student of Richard and mine, Mandy Truman, has developed for her study of modified vegetation on the Galapagos. So on the left-hand side is the historical sort of pre-disturbance state, and on the right-hand side a scattering of modified vegetation because of invasive species. And the red lines there are Mandy's expert opinion on where the thresholds might prevent the transition back to the historical state. And Mandy's going to talk some more about her work on Thursday if you're interested in seeing that. Data to prove the existence of thresholds is usually more less frequent, less common. This is some data that Vicky Kramer, a colleague of mine, collected to identify the threshold associated with the development of salt-affected vegetation in southwest Western Australia. She tracked the health of trees along a transect and related that to distance to the salty water table, where widespread clearing of the vegetation for agricultural development has led to a raising of that water table. And this is one of the rare examples, I think, of a true threshold in that it would be very hard to reverse back to that healthy woodland from the salt-affected woodland once the transition has occurred. Perhaps our biggest contribution to ecological theory was to suggest some new models for oldfield succession. So we're back in the wheat belt now. This figure shows the sort of classical model of oldfield succession, where once abandoned, they track back towards the pre-disturbed state along a broadly linear trajectory. We trawled the literature and found evidence for a second model, which passed through this hybrid ecosystem on the way back to the historic one. And thirdly, our data suggested the existence of a third model where the system basically got stuck in a degraded state and didn't pass back to that pre-disturbance historic system because of the existence of ecological thresholds. Okay, the third area I'm going to look at is how restoration practices helped us to explore the link between biodiversity and ecosystem function. And this work is really been invigorated by the contribution of Chris Holvey, shown in this photo here. She arrived to Richard's lab fresh from PhD with Erica Zavaleta, where this literature was very much part of her PhD work and she understood it very well. And essentially the literature tells us at the moment that this is the sort of saturating relationship between biodiversity and ecosystem function. And if we're interested in multiple ecosystem functions, then the available literature, and there isn't very much of it, and it's limited to grasslands, also suggests a saturating relationship between biodiversity and ecosystem function. And this was really the theory that we set out to test in a large-scale field experiment at Ridgefield, the farm I mentioned, where we planted trees in different combinations to really explore this idea. Our basic premise was that if we increased the species richness of those plantings, then we might expect higher levels of functions for multiple functions, including carbon sequestration, invasion resistance, pollination and biodiversity. And this is largely the work of Mike Perring, who's the lead postdoc on this project. And Ridgefield has just joined a global network of very similar experiments, which will of course increase our ability to generalise about the links between biodiversity and ecosystem function. And just a quick bit of evidence, we don't have any evidence from Ridgefield yet, it's too young, but Peenyup is 5 years old now, and we've just collected some data that suggests that both within and among these different vegetation associations, there is a positive relationship between species richness and above ground carbon storage, which is of course a very positive message to tell our policymakers and others who are interested in both carbon and biodiversity outcomes from carbon plantings. Finally, the outlook to the future. Perhaps the biggest challenge, I think, for my generation of ecologists and the next generation is to understand the impacts of global changes on our native ecosystems. So it seems logical to ask what restoration practice might have to teach us about these. And a bunch of colleagues and I have just been searching around for the right concept. We think resilience is the right concept to help us think about these changes, and we hope this paper will convince you of the same thing. In it, we've suggested that connectivity, scale, functional redundancy and response diversity might be important measures of resilience. And again, there's the potential to test some of these in restoration. So for example, Peenyup is part of a large-scale initiative to connect the Great Western Woodlands and the coastal forests around Margaret River. And there's the potential to buy up bits of farmland and do that restoration in such a way that we can test the contribution of connectivity to resilience of these systems to drought and fire and other disturbances that occur in these landscapes. Of course, more generally, ecosystem transformation is the norm, and we heard a little bit about this from Jorge. Current estimates are that up to 36% of terrestrial and 41% of marine ecosystems are novel combinations of new and old species, and so the question is, what impact is that having? My colleagues and I have started to think about what these compositional shifts might mean for function. So I've just drawn the historic system in blue there and the novel ecosystem with the pink being the novel species, but with the same function as the historic system, so there's no function or consequence there. We might imagine a scenario where the novel species have different traits, and so there's a difference in the frequency distribution of traits like seed size, plant height, and that sort of thing. In this case of species loss, we might expect a shrinking of the expression of function. And similarly with gain, we might expect an expansion. So a very simple way of looking at it, but again, there's the potential to test some of these ideas in restoration settings. And in brief, this is just some work that Bridget Johnson, a PhD student of ours, is doing to understand the effects of exotic species on plant pollinator networks. And these black bars, each of these black bars is a species, and she's drawn networks there. And that's just her collecting some data to understand if the visitors are actually pollinators. So in conclusion, I think that restoration settings have proved to be good model systems for understanding community assembly, ecological thresholds, and the link between biodiversity and ecosystem function. And the work's certainly not finished there. There's plenty of scope to continue with that. And I think they have potential to help us understand the ecological effects of global change. I'd like to thank these people, and particularly Richard Hobbs, for supporting my research for the last 10 years.
Thanks, Rachel. We have time for just one quick question on the floor. All right, well, but there'll be plenty of time to ask Rachel questions through morning tea. I should just also remind you that to apply for this award, there will be some notices put up in the bulletin and also the website for probably early in the new year. So before we go to morning tea, could you once again thank all the speakers this morning for some great and intellectually inspiring research.